Welcome to our program today. We're very excited to have with us four very important scientists in the space. We're discussing glucose regulation of beta cell KATP channels. Is a new model needed? Just to sort of give an overview of this discussion, we're going to have introduction and bios, and then we're going to follow with Doctors Rutter and Sweet talking to set the stage with the canonical model. And share a few slides. And then the Dr. Merrins and Kibbey team will introduce their model, share a few slides. Then there'll be a discussion between the two groups and then open discussion moderated by myself and my colleague, Alexis Carmer. Just a little bit about the speakers. Dr. Guy Rutter is here with us from University of Montreal. And after studying for a PhD in Bristol with Dick Denton, FRS, on mitochondrial calcium signaling, Dr. Rutter subsequently worked on the pancreatic beta cell as an MRC traveling fellow with Klaus Wallenheim in Geneva. They're working collaboratively with Tullio Posen and Rosario Pizzuti in Padua. He established new approaches to exploring the control of insulin release at the single cell level through the use of recombinant target probes and other techniques. He then returned to Bristol, becoming a full professor of biochemistry, and moved to Imperial College in 2006. His current research is supported by Wellcome Trust Senior Investigator and Royal Science Wolfson Merit Awards by the UK Research Council and Diabetes UK grants, as well as the European Commission Innovative Medicines Initiative in Diabetes. Prizes and award, he was awarded the Minkowski Prize of the European Association for the Study of Diabetes in 2004, the JRF Mary Jane Krugel Award in 2005, the Royal Society Wolfson Research Merit Award in 2011, a Wellcome Trust Senior Investigator Award in 2012, and the Dorothy Hodgkin Lectureship of Diabetes UK in 2014. Dr. Ian Sweet received his PhD from the University of Washington in bioengineering in 1993. He received the postdoctoral training in biochemistry at UPenn in the lab of Dr. Franz Machinsky until 1996. His laboratory focuses on the metabolic basis for disease, with particular emphasis on diabetes, cancer, and cardiovascular disease. The research has utilized sophisticated methods that have been engineered to non-invasively assess energetics and function in a diverse variety of cells and tissues important to these diseases. The wide range and applicability of the experimental approaches and research have resulted in funding from multiple sources: NIH, NIDDK, NSF, JDRF, Merck, and Seattle Foundation. As director of the Cell Function Analysis Core of the Diabetes Research Center, the methods developed by Dr. Sweet's group are available to other researchers as services carried out by 2 full-time technicians in the laboratory. Dr. Matthew Merrins, coming from the University of Wisconsin Madison. Research in the Merrins laboratory centers on the control of insulin release from the endocrine pancreas cells in Langerhans and how this is disrupted in diabetes. Their main interest lies in 2 features of nutrient metabolism in islet beta cells. 1, the ability to trigger pulses of insulin release, and 2, the ability to trigger cell proliferation when the demand for insulin increases, i.e. during aging and obesity. These adaptive responses to environmental stress ultimately fail in diabetes. To understand how this occurs, they utilize rodent models of obesity and aging in combinations with biochemistry, patch clamp electrophysiology, and quantitative imaging. A central focus of the lab is the use of FRET, optogenetics, FLIM, and 2-photon to monitor biochemical reactions as they occur in living cells. Our recent work is focused on the design and utilization of biosensors useful for real-time measurements of glycolysis, as well as the development of NADH FLIM, as a non-invasive optical approach to study the TCA cycle and ETC. Using these tools, they've been able to monitor metabolite production and second messenger signaling in a variety of pathways. And last but not least, Dr. Richard Kibbey, he's an MD PhD. He obtained his undergraduate degrees at Trinity University in San Antonio. He then obtained his MD PhD, University of Texas Southwest. His PhD was in cell and molecular biophysics and determining the NMR structure of peptides from the LDL receptor under his mentors, Drs. R.G.W. Anderson and R.L. Gierschach. Subsequently, he went to Yale, where he was selected for the ABIM short track in categorical internal medicine. In 2002, he stayed on for his endocrinology fellowship at Yale and is now board certified in internal medicine and endocrinology. And while in his fellowship, he worked in the laboratory of Dr. Gerald Schulman on metabolism and the pathophysiology of type 2 diabetes mellitus. And it was here that he identified mitochondrial GTP as a metabolic signal in the mitochondrial sensing flux in the pancreatic beta cell as a crucial component of the signal to secrete insulin. His lab has also developed a novel platform using stable isotopes and mass spec named Mimosa that measures the flow of metabolism inside and between tissues. And now he's an associate professor in the departments of internal medicine, endocrinology, and cell and molecular physiology. Okay, so thank you very much for joining us, esteemed scientists. And we have a quick overview for those of you who have not seen sort of these new diabetes papers that have come out recently. So Dr. Merrins and Kibbey put forward their paper, and this paper was titled "Glucose Regulation of Beta Cell KATP Channels. It is time for a new model." And sort of the thumbnail sketch is that maintenance of euglycemia relies on beta cells to couple nutrient sensing with appropriate insulin secretion. Contrary to what's often believed, the glucose signaling process in beta cells has not been solved, and the entrenched model implicating mitochondria in the KATP channel closure has been dismissed by recent genetic studies. To fill this void, we've proposed an alternative model in which glycolysis locally regulates KATP channels, and we'll discuss the strengths and weaknesses of this new model and propose experiments to test it further. And there was a second article by Drs. Rutter and Sweet, and their sort of thumbnail sketch of that article, well, the article was called "Glucose Regulation of B-cell KATP Channels, Is a New Model Needed?" And the thumbnail sketch of that article is that the so-called canonical model of pancreatic beta cell KATP channel regulation by ATP produced by mitochondrial oxphos is supported by a substantial body of self-consistent biochemical imaging, pathophysiological, and genetic data over the last 40 years. A new theory suggesting a role for the microdomains of ATP generated next to these channels by the glycolytic enzyme pyruvic kinase rejects the most parsimonious interpretation of these data. We argue that evidence provided in its support is incomplete, subject to the alternative interpretations, and as yet uncorroborated beyond its chief protagonists. While stimulating debate around the many as yet unsolved and important questions of the nutrient sensing coupling in beta cells, we argue that new findings fall a little short of dismissing the canonical model. And then I would also direct you for further reading to the Leslie Satin Sherman paper, which kind of brought this all together and urged these ideas not to be thrown out with the bathwater. So without further ado, I'm going to have Dr. Guy Rutter kind of, start off this conversation and, bring us up to speed with what's what.
Okay. Very many thanks, Monica. Let me share my screen if I'm able to do that. Okay, wow. Thank you so much, Monica, for the invitation to participate in discussion with the rest of the Sugar Science team. It's a great pleasure to do so. As time is quite short, what I thought I'd do was to channel Martin Luther and nail my theses to the church door right from the get-go. And I'm going to come up with just 4 key messages, and then Ian in a few moments will extend those. So what I'd like to tell you is that, first of all, the canonical model proposes that the stimulation of insulin secretion and, of course, the closure of ATP-sensitive potassium channels involves the substrate-driven oxidative phosphorylation, which chiefly produces ATP within the mitochondria. And I will argue that the configuration, the metabolic configuration of the beta cell, means that an independent control of glycolysis is very difficult in this setting. If you consider that a little bit too circumstantial, let me demonstrate the experiments which have been done to interfere with this process using inhibitors, using pharmacology, and then move on from that to explore experiments of nature, human genetics, and additionally mouse genetics. And then, with their indulgence and forgiveness, I hope, I will try to give a little bit of context to the alternative model, which will be presented by Drs. Merrins and Kibbey in a moment, to discuss some of the concerns we have around the ideas concerning microdomains of ATP close to the channels. Okay, so this is the canonical model, as we all know and love it. And essentially, what is proposed to happen is that an increase in glucose in the bloodstream flows through the glycolytic and the glycolytic pathway and the citrate cycle, and through the activation, the substrate-driven activation of the respiratory chain and oxidative phosphorylation, we see an increase in the production of ATP by the mitochondria, as well as a much more modest increase in the output of ATP in the glycolytic pathway. And these two contribute, although the role of mitochondrial ATP hugely predominates, in the closure of our eponymous friend, the ATP-sensitive potassium channel. And it's the subsequent electrophysiological events, the influx of calcium, which will ultimately trigger the exocytosis of insulin. Now, students of the glycolytic pathway will quickly point out that one of the steps in the pathway, glyceraldehyde phosphate dehydrogenase, will reduce NAD, and you need to reoxidize that NADH if the pathway is going to continue to flow. And Mike McDonald, a number of years ago, and we subsequently confirmed those data, pointed out that a glycerol phosphate shunt is very vigorous in the beta cell and takes care of all of that required reoxidation of NADH. Indeed, this is the only game in town in the beta cell, because the levels of lactate dehydrogenase, these are data from purified rat beta cells compared to alpha cells or liver cells, tell you the activity level that the capacity to produce lactate is extremely low. And we were struck when we did these studies with Klaus Vollheim many years ago, to the extent to which this differentiated feature of the beta cell was lost as we processed to less well differentiated and less functional beta cells. Moreover, the door to let out any lactate that you would make through that route is missing because monocarboxylate transporters, which are responsible for the transport of lactate and pyruvate, are another of the disallowed genes, as we and Franz Scheuch subsequently dubbed them, and this is completely absent of the protein and the mRNA level in the beta cell. So if you're thinking that a configuration like this, a kind of a pipeline in which the only place to go is for glucose carbon to flow through the respiratory chain, well, to flow through the oxidation by the citrate cycle is rather unusual, you'd be right. And you have to go as far afield in the biosphere to find an equivalent organism or cell type, which is so efficient at converting glucose carbon into CO2 and water, and that is insect flight muscle, which is, of course, all about producing huge amounts of ATP to produce the energy required for flight. And we demonstrated this highly efficient conversion of glucose carbon to CO2 and water in our first paper. This was independently, subsequently confirmed by Franz Scheuch and by Mark Prentke, and these are the two key numbers which demonstrate that about 90% of glucose carbon is fully converted to CO2. And this is a very different situation if you look at non-beta cells and alpha cells in this preparation. Moreover, this has functional consequences because highly or well-differentiated beta cell lines or purified beta cells have a very distinct response from almost every other cell in your body, and certainly less well-differentiated beta cell lines produced from the same insulinoma in the Vollheim lab. If we impose artificially anaerobicis by inhibiting oxidative phosphorylation with any of the inhibitors that I've indicated below, you see this classical Pasteur effect, this huge increase in lactate production as we get activation of PFK by citrate, the loss of inhibition by ATP. Lactate production by either primary beta cells or highly differentiated neuron cells is negligible to begin with and is eliminated when we suppress mitochondrial oxidation and the activity of the respiratory chain. Now, these drugs provide useful tools through which you could explore the functional consequences or the importance of oxidative phosphorylation in the control of ATP-sensitive potassium channels and insulin secretion. The other way of doing this, of course, is simply to add a protonophore, such as FCCP, and dissipate the proton gradient across the inner mitochondrial membrane. And these are experiments which were undertaken by Andrei Tarasov when he was a fellow in our group a few years ago, and I think he's on the call today. But the first thing that Andrei did in these rather sophisticated experiments based on single cells and combining electrophysiology with imaging was to show that the first event, when you report multiple parameters simultaneously, is an increase in the ATP to ADP ratio. Now, this is important for my argument because, as Ian will develop in a moment, this occurs simultaneously with an increase in oxygen consumption. And the only way that those two things can happen simultaneously is if it is the substrate, in this case pyruvate for mitochondrial oxidation, which is driving increased oxidative phosphorylation and ATP synthesis. We then see this rather vigorous oscillatory activity, which needn't detain us for the moment, but I think we'll come back to it later when we think about models which explain these oscillations. But the key piece of data I did want you to see was that at the end, which is that when we uncouple mitochondria fully, we see a complete reversal of these increases in ATP to ADP ratio. So strong pharmacological evidence for a role for oxidative phosphorylation in raising ATP. And indeed, there are experiments that were done many years ago, and I'm delighted to see that Steve Ashcroft is on the call today because he did experiments with Philip Randall more than 50 years ago, where he used uncouplers to show that they fully inhibited glucose-stimulated insulin secretion. There were studies from Ian Dukes, although he drew different conclusions later in the 90s, which showed that inhibition with either antimycin A or rotentin also inhibited glucose-stimulated insulin secretion very profoundly. And importantly, this is because we would argue these maneuvers by lowering ATP will within seconds increase the open probability of the ATP-sensitive potassium channel. And you can do this either with uncouplers or, as Satin recently reported, using, again, inhibitors of oxidative phosphorylation like azide. Okay, well, if that's not to your taste, if this is all too pharmacological, what about some genetics? What about the experiments of nature? Well, these are abundant. If we look at humans, for example, there are rare, highly penetrant forms of diabetes which are maternally inherited. And these are loss of function mutations in the transfer RNA, which is important in synthesizing some of the components of the respiratory chain. And one can identify cohorts, either in the Netherlands, in the UK or elsewhere, in which these mutations perfectly co-segregate with the disease, strongly indicative of causality. You can find less penetrant, but more common variants in other transcription factors, which again are associated statistically with diabetes risk. And this is a report from Charlotte Ling and Hendrik Mulder a few years ago. It's not quite genetics, but if you look at the level of mitochondrial proteins in islets from people with type 2 diabetes and compare to those of people who are normoglycemic, you see that there's a substantial lowering of the expression of a lot of mitochondrial proteins. So that's the human. What about the mouse? Well, here one's cup really overfloweth, and I don't have time to unpack all of this. But in summary, almost anything that you do which will inhibit oxidative phosphorylation will also lead to impairments in insulin secretion, and eventually, and mice do have means of compensating, will eventually lead to dysglycemia and in some cases to diabetes. And this is just one example which I wanted to use to illustrate the point. The details of this needn't detain us for very long. This is disrupting a mitochondrial protein involved in the fusion of those organelles. And what I want you to see is there's nothing subtle about this, but over time, you see this profound hyperglycemia, which results as a result of the deletion of this protein. And if I point this out, it's because these studies were performed in the UK, but in parallel and unbeknownst to us, almost identical studies using a different Cre-Deleta strain were being undertaken by Scott Soliman-Poor in Michigan with exactly the same results. So again, I emphasize the reproducibility and the robustness of these findings. Now, very briefly before I hand over to Ian, let me point out some aspects of the model which Drs. Kibbey and Merrins will point out. They will argue that a very localized domain of ATP production close to the channels will close those channels, and this is important and is the way in which the system works. And they are right to propose this insofar as this is what appears to happen, for example, in cardiomyocytes. And this has been demonstrated by several groups and confirmed in a recent study from the Satin lab, where you can see phosphenyl pyruvate will very nicely close ATP-sensitive potassium channels. The same group, using the same rig and the same solutions, had much more difficulty, unfortunately, in reproducing the same findings when they took equivalent patches from mouse beta cells. I'd also point out and Andrew Tarasov is also on the call, I believe that the concentrations of phosphenol pyruvate that we used were vastly in excess of those which pertain in the bulk cytosol of the beta cell. Okay, so my conclusions are that the canonical model of KATP channel regulation is supported by 40 years of internally consistent biochemical electrophysiological modeling, I didn't talk about that, imaging, mouse and human genetic evidence. And I would suggest that any model which is to overturn this would need to demonstrate critical flaws in the arguments, would also need to demonstrate the existence of robust mouse models, would also need to demonstrate, I think, that there is really genuine biochemical evidence for an association between pyruvate kinase and ATP-sensitive potassium channels using typical standards of proof, immunoprecipitation, and so on, and preferably show us robust human genetic evidence. And at the moment, those four elements are only partially completed. So with that, if I may, Monica, I will stop sharing and hand the baton over to Ian.
Yes, please. That was great. Thank you. Looks good.
Okay. Yeah. Okay, so I'm going to start in a way reiterating what Guy had started with, which was a diagram of how the metabolic control of critical signals comes about. But this is from the Banting lecture of Franz Machinsky and represents, as I said, 2 or 3 decades of research on how this occurs. And I'm going to use this figure to give a summary of what the 3 points I want to make. And so just to orient everybody, this is, we know glucose has to be metabolized. First traverses glycolysis, and then with 3 different pathways can stimulate ATP production. One is from the glycolytic pathway itself. One is about 5 ATP, depending on assumptions, from the hydrogen shuttles, which then traverse the electron transport chain, as well as the bulk of them coming from pyruvate and entering into the Krebs cycle and electron transport. So these ATP molecules then in this model will determine, along with usage, how much ATP and ADP is there. And as we know, then this can close KATP channels, increase calcium, and increase insulin secretion. So the real crux, I think, of the debate is what's going on with the mitochondria and its production of ATP within all this. We agree on most everything else that's shown here. I'm going to talk about 3 points, aspects of this, which will hopefully differentiate between our 2 models. And okay. The first point is one that Dr. Machinsky was well known for and got the Banting lecture on this, which was the understanding of how glucokinase acts essentially as a check valve. It is an initial step in glycolysis. Under the conditions where glucose transport is very fast, then glucokinase is able to sense essentially glucose in the blood. And by the position of glucokinase and the fact that it's a unidirectional flux rate limiting step, then the steps downstream act as a pipe. So there's, unlike other hexokinases, there's no feedback inhibition by glucose 6-phosphate. So whatever comes in is determined by glucokinase, and then the rest of the rates are simply keeping up with that rate that's determined by glucokinase. And so there's a control strength that's very large from glucokinase on the not only the production of ATP from glycolysis, but also from the shuttles and to the entry of pyruvate. As Guy pointed out, most of the glucose is oxidized to CO2. So the whole thing is kind of a pipe. And so in addition to talking about glucose and what went into that research that supports the model, I'm going to talk about, I think, an important point that the ATP that is generated from the mitochondria is actually substantial, and these are not, the mitochondria is not uncoupled. So we want to make sure that we want to show that the oxidation and processes in the mitochondria actually produce the ATP that's theoretically thought to happen. And then finally, to show that this ATP has access to the KATP channels rather than a microdomain will show its effect on insulin secretion. So those are the 3 points I'm gonna consider. I'm gonna start with one slide on the dominant control strength of glucokinase and on glycolysis as well as insulin secretion. And so this is a hugely important slide to the concept that glucokinase is the rate limiting step. And this shows here that the glucokinase dose response to glucose as well as the glycolytic rate as measured by glucose usage. And because they're superimposable, clearly the control strength of glucokinase is near 1. And that precludes, at least in a steady state fashion, of downstream processes controlling the rate of glycolysis. You can upregulate pyruvate kinase, but if no more comes through the check valve, it's not going to have impact. So it also is what establishes glucokinase as the glucose sensor because it's a high Km glucokinase, which then allows the beta cell to be sensitive to glucose changes in the blood in the physiologic range, unlike hexokinase. So I'm just going to, there's a lot of data I could show. I'm just going to show one brief experiment that we did in my lab with a glucokinase activator. And basically, we did this on a model of rat islets that had low glucokinase. We cultured the rat islets at low glucose. There's a glucose promoter on the glucokinase. So these islets then were 40 hours in low glucose, and that didn't change the response to a fuel that goes into the TCA cycle. We get insulin secretion rise, get a rise in the number of electrons flowing through the electron transport chain, and increases in oxygen consumption. And if we follow that, as expected, the response to glucose is very much lower because of glucokinase being lower. But as an example of showing that the control strength of glucokinase, we give an activator, and immediately the reductive state, the electrons flow through the electron transport chain as allowed by the increase in this gatekeeper. The oxygen consumption goes up, and we see in parallel rescue of the insulin secretion. And my guess is if you gave a pyruvate kinase activator, you would not see this because it can't possibly result in an increase of the glucokinase, which is needed to stimulate that process. And then I think, this research in my mind, one of the reasons I went to study with them is this, to me, it's sort of an apex of accomplishments in the diabetes field from this data that Dr. Machinsky and others predicted that glucokinase would have a large effect in diabetes. And so in 1992, it was a result of a number of studies in France. They've discovered that MODY patients, maturity onset diabetes of the young, are diabetic because of different mutations in glucokinase, which is definitely showing, which is obviously strong evidence in vivo that glucokinase indeed is showing this huge control strength. And a follow-up to that was they were able to relate, this is a model of the mutant human glucokinases from the different patients. And so going to the left here, you get more glucokinase activity than normal. And then this shows the threshold of the glucose response in patients. And so you can see that some of them even have hypoglycemia because the mutant caused an increase in glucokinase activity. So basically, we're showing a huge amount of overwhelming support that glucokinase is the major and dominant control element in both in vitro and in vivo. Okay, so I guess that's okay. So moving on, the second step, I want to show that glucose-stimulated respiration actually increases ATP, and it's not uncoupled. And so we did some very straightforward studies that are commonly done, and shown that here's rat islets. We start with the baseline at 3 millimolar glucose, and we're concerned mostly with the amount of ATP being generated by the glucose stimulus. And you can see here that when you give oligomycin, the blocker of the proton, channel that you completely block the oxygen consumption and therefore the ATP production that's occurring from glucose. And in agreement with this, you can take the mitochondria, in this case from beta HC cells, and show that the standard kind of relationship between ADP phosphorylation and oxygen usage is met within the PO ratio of 2.4. So both these indicate a low level of uncoupling and a direct relationship between oxygen consumption and ATP from oxidative phosphorylation. And here's another point I wanted to make that I think is different, could distinguish between our two models, and that's our conceptualization of what controls oxygen consumption. And here I'm drawing from Dave Wilson and Maria Erichinska, who were at UPenn when I was there, although they were there 20 years before me, and they've done dozens of papers and explained to me how the oxygen consumption is actually controlled not only by the ADP, which we're all very aware of, but it can be pushed by mass control by increased NADH. So to understand a change in oxygen consumption with respect to the mechanisms that mediate that, you need to know one or the other or both. It's very difficult to measure ADP. So we actually set up a system that could measure cytochrome C, the reductive state of cytochrome C. And this allows us to distinguish between push and pull by when we measure a change in oxygen consumption, as we've done here, the cytochrome C reduction went up. In other words, we measure the flow of electrons increasing. If this was going to happen by ADP, then you would see an increase in oxygen consumption would result in a decrease in the amount of electrons in that pool. So this is strong evidence that like most cells and most mitochondria, there's a dual control, and the beta cell certainly can increase oxygen consumption and ATP production while ATP has risen, as Guy had said. And to make this point very briefly, we did experiment where we took islets and we used them in our perfusion system overnight, so 20 hours, and they're still very sensitive to the amount of glucose. So even after 18 hours, there was no indication of fatigue or ATP buildup over 18 hours. It's very constant. It's a push system. You give them gas, they rev their engines, and they produce ATP. That's the only way it can really work if you want to understand how the glucokinase glucose sensor works, by having the metabolism in the beta cell transduce the dose response that we know is imperative to understand how an islet senses glucose. So on to my third point, which is, okay, the ATP is definitely generated by respiration in large quantities. Does it have access to the microdomain, or is there a microdomain? And so I'm going to present data that I actually did for another study, which was an attempt to identify which pathways were contributing to control steps in the insulin secretion. And of course, we looked at three different, it's a little bit different orientation, but glycolysis, TCA cycle. So the metabolism is going up here and then impinging on the steps that we're familiar with in terms of both models. But we used fuels that would enter into different parts of the pathways. So glucose is almost not really that useful in some sense because everything goes up. So it doesn't distinguish between mechanisms. So then we added KIC, which would then not include glycolysis, and attempt to see what steps were essential. And finally, we use glyceraldehyde, which actually, as I'll show, enters used through the glycerol phosphate shuttle and directly gives electron from FADH into cytochrome C. So the way you can distinguish between these two, as I'll show, is NADH. These two, of course, will both result in increases in NADH, and glyceraldehyde won't. And so let's see what this tells us. So on the left is the KIC responses, and on the right is the glyceraldehyde. And I didn't show glucose because essentially they're essentially the same as the KIC. If you go to 20 millimolar glucose, as I say, you give these compounds and all the important parameters go up. And so when you give glyceraldehyde, it was certainly know that insulin secretion also goes up, but we see similar calcium kinetics and responses. Oxygen consumption go up, reduced cytochrome C goes up. All these are a little less, and I'll explain why. But the NADH, this is the key, is it goes down instead of up. And this can be understood if we look at the glycerol phosphate shunt, where that takes it takes NADH and converts it to FADH, and then that eventually goes to cytochrome C. So there's a transfer of NADH to during the shuttle activation. But the moral of the story then, that given there's an increase in calcium in ISR, there's the inescapable conclusion that the ATP produced mitochondria has access to the KATP channel. And clearly there's no involvement of pyruvate kinase here with glyceraldehyde. So if there was any one piece of data I would point at to say this is what the new model needs to address, this would be it. And when I've gone for models, I've always tried very hard to find not the models that necessarily support my theory, but ones that can discredit it. We can have 50 experiments that support one, but only one is needed if it's valid to discredit a hypothesis. So I think this is a very important one that I'd like to hear their response. So in summary, I've tried to go over, review the data that shows the intrinsic mechanisms of a beta cell as a push system and as essentially a pipe with coupled mitochondria. And this ATP generated by OXPHOS is very large, doesn't turn off, and has access to KATP channels. And it's needed, downstream effect is needed fuel is needed to explain the effects of all fuel secretagogues, including glyceraldehyde, that is not expected to increase pyruvate kinase. And made the point that glucose is a unidirectional gatekeeper of metabolic rate that confers the glucose sensitivity in the physiologic range, and that the control strength has been shown high in vitro and in vivo, which in terms of the pipe model precludes downstream steps such as pyruvate kinase as controlling the overall flux through metabolic pathways in a steady state, although it could have a role in transients. And just to finish by saying, none of the data that I presented requires alternative models or the existence of microdomains of ATP and ADP. Thank you.
Great. Thank you very much for sort of team one, Drs. Rutter and Sweet, for presenting the historical data as well as their data backing their supposition. And so, yeah, can we now have Dr. Merrins and Kibbey introduce your model?
Yeah, sure. Thanks so much, Ian, for teeing this up. I thought I'd start with a quote from the scientific philosopher Karl Popper, who says, "Whenever we propose a solution to a problem, we ought to try as hard as we can to overthrow the solution rather than defend it." And it is in that spirit that we're going to undertake this talk. Popper also says that it's really important that we state our problem as clearly as we can and put our solution in a sufficiently definite form where we can discuss it. And so I hope that the researchers out there will forgive us for boiling down many elegant studies into these simplistic models. But in its simplest form, the canonical model, which is shown on the left, is that oxidative phosphorylation is fuel limited by glucokinase, as Ian said, and raises the ATP-ADP ratio in the cytosol to close KATP channels. And Dick and I have instead proposed that it's pyruvate kinase that's fuel limited by glucokinase and closes KATP channels. And so actually your arguments, Ian, about glucokinase really aren't germane. We agree on what glucokinase does. It is the glucose sensor for the beta cell. Pyruvate kinase cannot control the amount of fuel that comes in. So I'm going to try and get everybody on the same page so that we're having the same argument about the same point. It's really about this blue box right here. Which of these two ATP generating processes generates the ATP that closes KATP channels? And a lot of people are asking, and rightfully so, is this new model correct? And if we go back to Popper on the preceding slide, he would start with a different question. He would ask, can the canonical model be disproven? And so a really nice study came out last year from Anna Lang and Alejandro Caicedo's group, Miami. What Anna did is she knocked out 3 different respiratory chain complexes, specifically in beta cells, complex 1, complex 3, or complex 4. And in all 3 cases, she saw the expected reduction in oxidative phosphorylation, which in the canonical model generates ATP to close KATP channels. And in all 3 cases, she found that there was absolutely no difference in glucose-stimulated insulin secretion in the isolated islets. When they went deeper, what they reported was that in 2 of these models, there's actually an equal number of beta cells responding to calcium. There is a decrease in the amplitude of the calcium that comes in. But interestingly, in this most severe model, which is the complex 3, what they found is it actually takes less time to depolarize the cell. So right here, with 3 different models, any one of which you might be able to dispense, we can do away with the canonical model. And if you're not convinced by this, we can turn to another group, which is Scott Solomonpour's group. So Emily Walker, who's a postdoc in the, a scientist in the group, knocked out the mitochondrial phosphate transporter. This is the protein that brings in phosphate to be combined with ADP for mitochondria to make ATP. Yeah, here too, she reported decreased oxidative phosphorylation. And in this case, you see a decreased insulin secretion. The really interesting part of this model is that what they found is, with their collaborators in the SATIN group, that you see beautiful calcium oscillations at low glucose. So these are 4 different knockouts, and this is quite different from the mitofusin knockout, which, I mean, the mitofusin is really about fission and fusion, but these knockouts are a more strict test of does OXPHOS generate ATP to close KATP channels, and we get the same answer 4 times in a row. So I think to address the key question about this talk, do we need a new model? Yes, we absolutely do. And so I think a lot of people in the audience are asking now, well, if there is a new model, if we do need a new model, does our model hold water? And so we're going to talk about, in the brief time we have, 2 sort of key hypotheses that are separable. So the spatial hypothesis, which I'm going to talk about, is that glycolysis preferentially controls KATP channels because of its location at the KATP channel. But there's a second hypothesis, which Dick is going to talk about, and this is a bioenergetic hypothesis. And what this posits is that OXPHOS is unable to raise the ATP-ADP ratio to the KATP closing threshold. But pyruvate kinase can do this because of the favorable thermodynamics of the PK reaction. And just to clear up a misconception from the prior presentations, we are not suggesting that ATP is unable to diffuse from the mitochondria to some privileged microdomain where it just can't make the diffusion. That would be ridiculous, because if you just take a beta cell and you put holes in it, you can put ATP on the outside of a beta cell and it will close the KATP channel inside. So we're not saying there's anything funny going on with diffusion here. We're simply questioning whether mitochondria, due to their bioenergetic properties, are capable of producing ATP at the time KATP channels close. Now, many of us, when we see these recordings of oxygen consumption, after a step increase in glucose, we'll see that there's massive oxygen consumption. But I would like to remind the audience that stepping from 3 millimolar glucose from the starvation state is not physiologic in the least. That's a hypoglycemic condition. If we sit at 3 millimolar for an hour, we're all going to fall over. What the fasting state is, it's already 4 millimolar. So a lot of this oxygen consumption is just the cell putting back what we took away from it in the first place. It's an artifact of the system that we all use, where we glucose starve before we raise glucose to see insulin be released. Okay, so what about this spatial hypothesis? What's the evidence for this? So the first bit of evidence for this is biochemical. So if you pull down KAR 6.2 or SIR1, these are the two subunits of the KATP channel. You'll also pull down glycolytic enzymes, including pyruvate kinase. Now, this has been done by yeast 2 hybrid. It's been done by co-immunoprecipitation. And in these experiments, you can take a COS cell, not a beta cell at all, overexpress the channel subunits, and it will pull down glycolysis. And finally, if you prefer the endogenous proteins, there have been mass spectrometry experiments done where the endogenous proteins have been pulled down and glycolysis comes with it. And it's been stated, including in a review from Lassatin, that there is zero evidence for this in a beta cell, but that's just not true. And I'll just put the references down here so you guys can screen capture them. It's reviewed in, the data are reviewed in Foster and Coetzee, but here's the Min6 cell data that you'll be interested in. And so, would I feel better if we repeated these biochemical experiments in purified beta cells from humans? Yes. Will we do this? Yes. Have we gotten to it? No. Okay, but there is evidence. So we can't say there's none. That's not fair. The second bit of evidence is that pyruvate kinase closes KATP channels in excised plasma membranes from human and mouse beta cells. So this was work undertaken by 2 electrophysiologists in my lab, a grad student and a scientist. And what they did was they pulled small patches of plasma membrane from human and mouse beta cells and showed that the ADP and phosphoenolpyruvate application in this very contrived system will close KATP channels. We have repeated these experiments with micromolar levels of PEP. They are unpublished, but we see the same thing that we do with the millimolar PEP. We simply use the millimolar as a starting place because that's where the heart field started. And so we reproduce their experiments first. So a key part of this is that when we first did this, we really didn't understand what isoforms were present at the KATP channel. And this gets back to this genetic point. People keep coming at us and saying, why don't you see that there are defects in pyruvate kinase that causes diabetes? If you have a defect in pyruvate kinase, you die. You don't cause diabetes, you're dead. And so it's not like glucokinase. Glucokinase is also known as hexokinase 4. There's multiple isoforms here. And I agree with everything that Ian said about glucokinase. It's the glucose sensor. But in terms of the genetics, I think we don't see it because of this redundancy. So there's actually 3 isoforms in beta cells. There's a constitutively active PKM1, and then there's 2 allosterically recruited isoforms. And we actually undertook experiments where we actually knocked out the different isoforms, and this is published work. And what we found is that if we knocked out the PKM1 isoform, this is the constitutively active isoform, that we could block KATP channel closure. But there's a catch. And the catch is this is not the only isoform that closes KATP channels. If we simply take those same membrane patches and we activate the allosterically recruitable isoforms, which get pulled off with the plasma membrane patch, we immediately rescue and we close the KATP channel. Now, this experiment is contrived. It is effectively a biochemical experiment. And so we've also undertaken experiments in living cells that are intact. And here, to get around this redundancy, we had to use another approach because at high glucose, the 2 isoforms of pyruvate kinase at the channel are effectively equivalent because glycolysis will provide FBP to activate the allosterically recruitable isoforms. So in these experiments, we did measurements at low glucose with mitochondrial fuels. Now, what Ian said about mitochondrial fuels sort of ruling out our model is not the case. Mitochondrial fuels do not bypass the pyruvate kinase reaction. This we demonstrated in Foster et al. Just a couple of years ago. So if the key amino acid is leucine. And so leucine, if given to a beta cell, will work through the PCK2 enzyme to make PEP. And we argue that goes through pyruvate kinase to close KATP channels. And that's what we see in the experiments. So these are intact beta cells stimulated with amino acids. You can see there's KATP closure. We can knock out the constitutively active PKM1 isoform and block this. And I want to point out something very important about these experiments. We also did imaging experiments at the same time. And what we found is that the ATP-ADP ratio in these knockouts is equivalent to the controls. So that seems to argue that the ATP-ADP ratio in this bulk cytosol is insufficient to close KATP. And if just to sort of bolster our electrophysiology experiments, we also measured calcium and insulin. And just like the KATP closure was blocked, so was calcium influx and insulin secretion. So I don't think there's a part of our argument that rests so firmly on the electrophysiology data, because we can get different sets of hands with different assays telling us the same thing. But of course, if we reactivate the allosterically recruitable isoforms, we get KATP channels to close. And just since I know that everybody is a little bit over time here, I'm gonna sort of cut a little bit and just sort of summarize a little bit more of the data. We think that it's not just the pyruvate kinase enzyme, but there's actually a whole cluster of glycolytic enzymes. And this is all supported by the biochemical evidence that the Kutsi group had shown in Min6 cells. We find the same enzymes are attached to KATP in these excise patch clamp experiments. They show that they're attached. We show that they regulate the channel directly with electrophysiology. So what we've shown effectively is that there are two isoforms at least that redundantly close KATP channels, PKM1 and PKM2. And a very reasonable question, which Patrick Roersman asked at the ADA debate about this last year, was, could you show us what happens when you delete both isoforms? Because if you delete just one, the phenotype of the mouse is pretty much fine. And we did that. And you get diabetes. The mice get diabetes. So here are the data. Here's a glucose tolerance test. And here's the ex vivo insulin secretion showing a profound defect in glucose-stimulated insulin secretion. Probably my favorite part of this experiment is that if we reactivate the L isoform, which is barely expressed in beta cells, we can completely rescue the phenotype of these animals. And if you're asking yourself, why over here didn't the fructose bisphosphate from glycolysis just rescue? I would answer, well, maybe there are metabolic compartments. Now, I fully concede that we have yet to demonstrate that there are microdomains that matter. So in other words, if we move pyruvate kinase, does it still work? According to the spatial hypothesis, it shouldn't. And if you come to our ADA talk, we'll be imaging glycolysis in the microdomains of the cells. So this is a beautiful image from Jing Hughes' group, which shows you that this carpeted area, these are actually the microvilli, and there are primary cilia. And what my students have been able to do recently is image metabolism in these tiny little compartments. These compartments are much smaller than a patch pipette, and they're very active. And Leo Orci showed us in 1989 that this is where the glucose sensing machinery lives. So that ought to be fun. And with that, I know we're so short on time, so I'm going to just turn it over to Dick.
Fantastic. Really looking forward to that ADA talk. Really interesting.
Okay. Well, everyone loves bioenergetics, so I'll rush through it in the few remaining moments that we have. And just like Matt mentioned, he shares, we share a love of glucokinase. We also share a love of mitochondria. And we don't want to dismiss mitochondria from this picture because we do think that they're important and they have a functional role. And they do provide a basis of ATP within the cell. But boiling down the bioenergetic hypothesis to its simplest form, it suggests that oxidative phosphorylation in a beta cell can't raise the ATP to ADP ratio high enough to close KATP channels. As they could make all the ATP in the world, but it's all about the ATP to ADP ratio. And that's a thermodynamic issue. And we think that pyruvate kinase can do it, but OXPHOS can't. So in So basically what I'm going to argue is that beta cell mitochondria are hobbled. They're electron and carbon sieves. Beta cell oxphos turns off before ATP to ADP is high enough to close the KATP channel. Pyruvate kinase is a substrate responsive at high ATP to ADP ratios when oxidative phosphorylation is turned off. And pyruvate kinase has favorable energetics to close the KATP channel. These come from, in part, how it's made. It essentially takes 2 ATPs to make 1 phospholipopyruvate, which yields 1 ATP. So you're transferring biochemically a lot of free energy out to the cytosol through this reaction, more than what the mitochondria can do. Now, we heard a lot of arguments about stoichiometry and mitochondria and other things. I'm going to say there are a lot of irrelevant arguments relative to the hypothesis that we're proposing. The stoichiometry, even if the numbers were correct, which I would disagree with, we could talk a lot about those, the stoichiometry really doesn't have any relevance to this argument. I would also make the comment that a lot of the thinking that has gone into understanding the beta cell has come from composite measures, things like oxygen consumption. Oxygen consumption does not equal oxidative phosphorylation. It's more complicated than that. Also, NADPH autofluorescence does not equal mitochondrial NADH reduction. This is a really important question. I think we're going to be hearing a lot more about this in the next few months. But while NADPH autofluorescence is a useful biosensor to time things, it really doesn't have much to do with the mitoredox state. Finally, the spatial compartmentation arguments that Matt just made, they don't really relate to what I'm going to talk to you about, as well as PEP cycling, whether you like it, understand it, or don't like it. It has no role in this particular hypothesis except as an amplifier. And I'd just like to point out that, that in the beta cell, ATP is pretty much fixed the whole time within the physiologic range of the beta cell normal function. It's really the ADP that's changing. It's several orders of magnitude lower, and it has to go from low to extremely low. And that's a bioenergetic issue. It's not a kinetic issue. And that's really key to understanding what we're talking about. So the relevant questions are, is pyruvate kinase essential? And I think Matt just showed with a number of his studies that yes, it is essential. Another relevant question is, are beta cell mitochondria weak? Well, there's a really lovely paper by Effortit and Brand that shows, yeah, they're really weak. And they showed a number of interesting features about mitochondria and beta cell mitochondria in particular. So the ATP to ADP ratio is determined by the delta psi, or the proton motive force. It's not related to oxygen consumption. In fact, as the ATP to ADP ratio goes up, oxygen consumption goes down. That's a really, really important consideration when we think of this. Not only that, most of the oxygen consumptions in beta cell under approximately state 3 condition is leak. More than half of it is going to leak or to other metabolic processes besides making ATP. So oxygen consumption is a very poor surrogate. Finally, the maximum ATP to ADP ratio that a beta cell mitochondria can make is far lower than what muscle mitochondria can make because there's a large inherent difference in their ability of the mitochondria to generate the thermodynamic force to raise the ratio. Okay. So we heard that there is not a way to dispose of lactate outside of a cell, unlike other tissues. And that would seem to insist that glycolysis is fundamentally stoichiometrically coupled to oxidative phosphorylation. And that is true in many tissues like muscle, where pretty much all the ATP that's coming out is coming from oxidative phosphorylation. But beta cells are quite different. A large number of labs have shown a large number of alternative pathways that are very active in beta cells in response to glucose. For instance, lactate, citrate, and glutamate concentrations accumulate in the cells and then they go down. So they oscillate back and forth. So that allows more flux through pyruvate kinase that is not going into the generation of ATP. There are reappropriation pathways where electrons go into non-oxphos-dependent substances like NADPH. There's massive amounts of leak, like I just showed you in the mitochondria. Plus, there's the ability to generate peroxide that divert carbon flux. And finally, there are cycles that are really important. In particular, the PEP cycle, not only does it consume some of the mitochondrial membrane potential, but it also regenerates PEP in the cytosol and doubles essentially the contribution of pyruvate kinase. So we think that glycolysis is independent of oxidative phosphorylation. Now, can pyruvate kinase raise the ATP to ADP ratio higher than oxidative phosphorylation can? Well, let's have them compete. So what we can do is we can do a phosphocreatine clamp, fix the ATP to ADP ratio, and look to see at the effect at which one dies out before the others. And guess what? At around the range where high glucose is found, mitochondria activity is suppressed significantly, whereas pyruvate kinase is completely able to operate under those conditions. So that really says that oxidative phosphorylation is outcompeted by pyruvate kinase. And we can even show that in further detail. These are human islets where we can add phosphoenolpyruvate at physiologically relevant concentrations and show that it suppresses respiration related to oxidative phosphorylation in these human islets. So pyruvate kinase outcompetes the ATP to ADP ratio. But one of the most important components of the canonical model is that there is a substrate limitation of the mitochondria until the glucose is raised. Well, if that were the case, then the expectation would be that if you increase the amount of work by uncoupling the mitochondria, there should be no difference in respiration because they are substrate limited. And what you can see at 4 different glucose concentrations, as we slowly titrate in uncouplers, we see that not only is the demand effect far greater than the substrate effect, but that there's more than enough capacity within these cells without changing the fuel otherwise to respond to respiration. So there's no way that these are substrate limited. So with that, I return to our 2 models, the spatial and bioenergetic models. And if we ask, do we need a new model? I would say yes. And I would say, is our model correct? It's probably way too soon to tell. And I think some of the points that were made earlier about some of the additional work that needs to be done are entirely valid. But I think this is a really important step forward in a new direction. And with that, I will stop my share.
Thanks very much for that. You know, we've run a little over, but I think it's very important to have a discussion between the 2 groups. So let's say we have a few minutes, maybe 5 to 8 minutes for discussion between the 4 of you. And then we could if time, you know, if you're able, we could take maybe 2 to 3 questions. And as sort of a follow-up, we will have a submission piece in our next our Week in Review newsletter for people to submit questions, and then we'll have them answered in the following one, something along those lines. It was a really great discussion so far. So do you want to, you know, you have the ability to unmute yourselves and sort of crosstalk here if you'd like, or we can just go to questions. It's up to you.
Perhaps, could I say something about the Lang paper, since Matt's hanging an awful lot of that, the Caicedo paper?
Yes.
and ignoring all of the other manuscripts and papers that I presented. Fair enough. There is a non-equivalence between the deletion of the different complexes. Obviously, if you delete complex 1, there's a lot of opportunity for alternative substrates to stimulate the respiratory chain. Complex 4 is more complicated. That ought to have a profound phenotype. Complex 3 is probably the one that would have the strongest phenotype, and it does. And by the way, that's accompanied in that paper from Caicedo and colleagues by a huge increase in the expression of glycolytic enzymes. So glycolysis increases, and that doesn't compensate. What I would say also is that if you look carefully at those data, and having done, like our opponents, a lot of work in mice over the years, those animals do eventually show signs of developing frank diabetes. And the time courses over which those experiments were run were quite short, I think 8 weeks after tamoxifen deletion. The fact that you see an increase in diabetes at all, if you flip that back to human genetics, you'd be singing and dancing and saying this is a demonstration that that gene is important in disease pathology. So you can read those data as you wish. So that would be my first observation. Thank you to Dick for raising those points about lactate and glutamate accumulation. These are not wrong, but the key word here is accumulation. These substrates are not getting out of the cell, so the metabolic flow is extremely low. So I'm not sure quite how to understand those experiments really. So those are a couple of points that I wanted to make, and perhaps Ian might, if he may be allowed, to chime in with some responses to some of what Dick was saying about the bioenergetics.
So maybe we should not confuse everyone.
Oh, sorry.
Yeah, Matt, why don't you go ahead and respond to the Lang paper?
I think the Lang paper is the most direct argument against the canonical model that has ever been. It's directly targeting oxidative phosphorylation, and there's no defect in insulin secretion. I mean, that's full stop. We're done. The canonical model does not.
You may be done, but the animals do
experiment.
develop diabetes. And just to reiterate my point, if this were in a human setting, you would be announcing that you had identified a gene associated with diabetes. So, and as I said, I admire your presentation of the Popper model. Let's try to examine those data more closely. It's true the in vitro data are not terribly informative, but the animals do develop diabetes, or some of them do over time. So how do you address that? Do you dismiss that?
The complex 3, I mean, so the in vivo phenotype gets clouded by beta cell mass changes. But I mean, the real question is, how do these cells, which now have dramatically reduced oxidative phosphorylation, as demonstrated by, or at least as proxied by, I think is a fair way to say, the seahorse assays, how are they closing KATP channels? I mean, OXPHOS is absolutely crippled, and yet calcium is coming in. And in one case, the calcium
do we know what else is happening? Do we know what else is
faster.
happening to those cells? I don't recall.
Well, I don't know what's happening to those cells, but I know that those data, and so are the data from Emily Walker and Scott Solomonpour. You simply cannot knock out the mitochondrial phosphate transporter and have mitochondria phosphorylate ATP. It can't happen. So if it can't happen, but yet the KATP channels are closing, there's beautiful calcium oscillations. They're gorgeous, and they're at low glucose. This doesn't fit with the canonical model. We need a reevaluation. Maybe not everything
what is
about our
needed in
model
all
is
of
right.
biology is to be able to do experiments much more rapidly, where we can make these changes within minutes. All manner of compensations can happen. I could, give you.
But how would the
although I didn't
mitochondria
elaborate.
make ATP? That's the problem.
Well, the mitochondria wouldn't have to make ATP if there were compensations and other pathways and glycolysis and other pathways revved up, which is perfectly possible and happens vigorously in the complex 3 model. So it's difficult to interpret those experiments. But how do you, Matt, face off the other string of knockouts that I described and cellular experiments, row 0 cells, for example, where you have crippled your mitochondria, you have crippled your mitochondria and you lose secretion. So why is it disappeared in those settings? Can we be autocritical and sort of say, what else is going on there? Why does that work? Why do you lose secretion?
why do you lose secretion? I mean, I think that, you know, you and Ian addressed that nicely in your review. I mean, the same with the drugs. I mean, if you do block completely, you're going to slow glycolysis down. And so those experiments where you block the electron transport chain, they do impact glycolysis indirectly, and they don't really help us get between our models. I mean, sort of as Ian was arguing, the crux of this is, can we design an experiment that rules out one model without taking out the other model, right? And I think that the Caicedo data are pretty strong evidence of that.
Can you humor me
Whether
then and respond to the glyceraldehyde data, which we put in the paper and we presented now as a very nice experiment that would separate the two? Is there any involvement of PK and PEP from glyceraldehyde? I don't know there is. And so this is definitely an experiment that would separate out the two.
Why
it
not?
wouldn't, because glyceraldehyde's upstream of PK.
It enters into, it directly donates electrons to the glycerol phosphate shunt. So it doesn't traverse glycolysis or the TCA cycle. And that was the data I presented.
Dick, you want to take this one?
Yeah, sure. I mean, there are a couple points about glyceraldehyde. I was kind of surprised you actually used that as a strong argument, especially because you showed in your paper that if you inhibit complex 4, that it has no effect, that it's all cytochrome C that was important.
I didn't do that. Wait, we got to make sure that's not the case. Don't.
No, that was a
things that I
TMP
didn't do.
ascorbate experiment in the paper.
That didn't
But
block anything. That was a stimulator, and it didn't make insulin secretion. So make sure
exactly.
quote me right.
Exactly. Okay. But you haven't shown that the glycerol doesn't get phosphorylated, or the glyceraldehyde doesn't get phosphorylated. Pete Antonazzi did show that he was able to get those compounds through the metabolism. And so again, what's going on with the glyceraldehyde experiment, we don't entirely know.
Well, if you said that what you said was right, you would have NADH production. Glucose makes NADH go up, glyceraldehyde doesn't. So clearly it's not traversing glycolysis.
No, you didn't measure NADH. You measured NADPH, P in parentheses. You don't know what's going up and what's not going up in that experiment. So it really becomes a struggle to interpret that. And that's why I started out with. I mean, that measurement is just so unhelpful in understanding the bioenergetics of the system. And Matt can maybe throw a little bit more.
I think just because it has a mixture of signals doesn't mean it doesn't tell us something.
It's worse than that, Ian, actually. So we've gone back and looked at mitochondrial NAD levels and the mitochondrial NAD to NADH ratio with biosensors from different groups. And in both cases, when calcium is high during the active phase of oscillations, you see something quite different from the autofluorescence and the biosensors. And so the reality of those measurements is autofluorescence is a number of different mixed signals. And it's not just NADH and NADPH. There are other signals in there. There's a whole community around, a lot of it's at UCLA actually, that has been looking at, and UCSD, that has been looking at what those intrinsic autofluorescence signals are. And unfortunately, they're contaminated during that phase. And so that really muddies the waters. And so they don't match.
Sure.
So I don't think we can use that.
Even if it's a mixture of signals, it goes up with glucose and it goes up with KIC. So we know that the production of highly, of electrons that are going into the electron transport chain are reflected by that. So if you think there's some contaminating signal that is eliminating that signal from glyceraldehyde, well, that's a far-fetched. So I think, yeah, sure, it is a bit muddy, but that's not a reason to say, well, we can ignore it when it says such an important thing that could distinguish between. I think you have to answer to it and not ignore it.
I mean, I think the reality is this, the idea that mitochondrial fuels close KATP channels and initiate calcium influx has been known for a long time. And we can show that at low glucose, that will not happen in the absence of mitochondrial PEP production by PCK2. It will not happen without an active pyruvate kinase at the KATP channel. And so those are two genetic experiments that provide an alternative explanation for data that's well known. I mean, the canonical model says that the KATP channels closed because you gave mitochondrial fuels and that mitochondria made ATP to close KATP channels. But the genetics tells us otherwise.
We're now at the 10:17 mark, and I just wanted to, give accolades to everyone in the discussion, and also just kind of reference the fact that science is built, on the backs of all who came before, and perhaps this is maybe this point of inflection is the result of all these new tools that are now available, and building on the great science that's already been created are these new hypotheses that come along with new tools. So I just wanted to sort of add that, and I also wanted to say that we have many questions. We have many pre-submitted questions, and we really have run out of time. So as an offering, instead of taking the questions now, we'd like to propose that we have an opening in our WEIR, the Week in Review, which we put out tomorrow, for people to ask further questions and for us to send, collate all the questions we have, and then we'll submit them to the 4 scientists, and perhaps they could respond, and we'll publish that in the following week. And in that way, we can entertain people's questions, if that's okay. But I really do think this is just such a fascinating and such an important discussion right at the forefront, right at the frontier of where things are and where they may be going. And I appreciate everyone's respectful, discussion and respect of each other. It's so important to have these kinds of conversations in this context. So thank you again, everyone. And with that, we'll say goodbye. I know everyone is so busy, and we will be in touch again regarding the questions and sharing them with our population. Our WEIR now reaches over 5,000 scientists globally. So thanks again, Drs. Rutter, Dr. Sweet, Dr. Kibbey, and Dr. Merrins. And thank you for championing the full understanding of the beta cell, which is so important in T1D. Have a great rest of your day. Bye-bye now.
Thank you.
Thank you.
Everyone.
Thanks, everybody.